Let's look into the enzyme which are commonly used in genetic engineering, that is RE, or it is also called it as restrictions endonuclease. ASE is referred to enzyme, endo means inside. This is referred to the DNA or the nucleus. So it is referred to the nucleotide. So it is an enzyme that is going to cut the DNA inside of the cells. So you have restrictions here. So DNA that has been cut, we call it as a restricted DNA. In nature, you can find this enzyme a lot in the bacteria. Let's say this is a bacteria cells. Let's draw a rod-shaped bacteria with a flagella here. Inside bacteria, they have their own DNA that is lies freely in the cytoplasm, and they also got one small circular DNA that we call it as a plasmid. Other than that, they also contain a lot of enzyme in their cytoplasm. Some of the enzyme is help in their cellular respiration to produce ATP in their metabolism, and some of this enzyme is help to prevent the invasions of the viruses. Let's say you have virus that attack this bacteria. Virus will inject its DNA into the cytoplasm of the bacterial cells. And this DNA is going to incorporate into the bacterial DNA, and it is going to hijack the machinery of this bacteria. And then it will use as a breeding ground for the viruses. Now, to prevent the invasions of viruses, the bacteria will have some mechanism to help to destroy the viruses. Now, some of the bacteria contain this one, which is called as RE or restrictions endonuclease, or sometimes it can be called as restrictions enzyme. The functions of this enzyme is they will cut the DNA into pieces, and they have one characteristic that is very specific, highly specific. Specific means that they only recognize a certain sequence of the DNA. The DNA of the viruses may be very long. DNA or RNA of viruses can be very long, and alongside of the DNA, they have a special sequences, and that is going to be recognized by the restrictions enzyme. So this restrictions enzyme is going to target the DNA of the viruses and cut into pieces. So it is going to screen along these DNA sequences. Let's say example here, you will recognize the sequence of GAATTC. So if you read through all the bases of the DNA here, when you see this point that has GAATTC, you're going to cut it. Another point that has GAATTC. So this cutting point, we call it as a restriction site. And they will form into, after cutting, they will form into individual pieces. So you have 1, 2, 3, 4, 5, 6 pieces here. We call these pieces as the restrictions fragment. So if a DNA sequences has 5 restriction sites, you're going to have 6 restrictions fragment. Same thing, if you have 7 restriction sites, then you are going to produce 8 restrictions fragment. If totally no restriction site here, the viruses is going to have 1 fragment of DNA intact, and this DNA can be used to incorporate it into the host DNA sequences. If this one have 3 restriction sites, that means you will have 4 restrictions fragments. Now we already know it is very specific, meaning that they only recognize specific sequence. And most of the sequence that you can find that is identified by the restrictions enzyme is palindromic sequences. Now let's look into this one. Let's say there is an enzyme that is going to cut the sequence A A G C T T, and you know DNA is double helix, and if you have A A, the opposite strand must have T T, so it is always double helix. So we are this is double helix structure, so we are unravel it into linear shape. So you have T T here, C G A A. Let's say this is 5 prime to 3 prime. The bottom line here should be 3 prime to 5 prime. It is always anti-parallel. This is one of the nature of DNA strand. There is an enzyme that will recognize this specific sequence. That enzyme name is called as HIN3. You no need to remember the enzyme name and also the sequence that is going to digest, but you should at least know that the digestions of the enzyme sometimes will produce blunt end and sticky end. Now we will look into this HIN3. It is going to produce sticky end by digest your DNA sequences of AAGCTT in this way. Now we will look into here, we call this as palindromic site is because if you read the sequence from 5 prime to 3 prime, it is going to have AAGCTT. While you read from the bottom strand from 5 prime to 3 prime, the sequence are exactly the same. It is AAGCTT. So we call this a palindromic site. All your different types of the restrictions enzyme, they will cut at this site. So restrictions enzyme is act as a scissor. It is going to cut the DNA and they will produce fragment. So in this case, you will have a fragment like this. And inside you will have nucleotide bases. So from one DNA sequences here, it divided into 2 fragments due to the restrictions endonuclease digestions. And this site we call it as a sticky end. Sticky ends allow the researcher to apply in cloning where they can put it a new DNA sequences that have the complementary sequence to this sticky end. Then you can incorporate a new DNA genes inside here to do cloning activity. Another one, other than sticky end, it is also can produce blunt end. Blunt end example, if you look into this sequence, you have G, A, T, A, T, C. And the opposite end will be C, T, A, T, A, G. So this is the code of your DNA. There is a few rules here where you will have A always bind with T with 2 hydrogens bonding. C always bind with G with 3 hydrogens bond. So this is how they form a helix, double helix string. Now in this case, you have another type of enzyme. We name it as EcoRV. That is going to cut the DNA vertically, and they will produce a fragment. So this is the RE digestion. So you will produce this type of fragment. This is GATCTA. This is ATCTAG. This is called as blunt end. Now the difference between these two, sticky end and blunt end, is depends on what type of the enzyme that used in the research or the experiment. If you tend to get the HIM3 or PSD1, you will get the sticky end productions. Now sticky end is not only they have double stranded here, they will have at the end of your DNA sequence, you're going to have a single stranded end like here. So this is all your single stranded end that is overhanging, protruding at the end of your DNA sequences. Blunt end, there are no single stranded DNA. You only got the double stranded DNA that is split into the two fragments. Now compare between these two, sticky end is better because the sticky end easily form the hydrogen bond with the complementary sequence on other DNA fragments that cut using the same restrictions enzyme. So if you're going to clone something, it will bind tighter because as long as your DNA ending, your genes ending has the complementary DNA sequences, increase the chance of the cloning process.